Okay, thank you everybody for attending. My name is Robert Owen. I used to be program director for Oracle. I was program director for Asia Pacific for Oracle Computers. And then several years ago, although they really paid me well, the amount of work they gave me really got on top of me. I was working all evening, all night, all weekend. So I spat the dummy one day and walked in and resigned. Now, needing something to do, I approached Melbourne University and suggested that I did a PhD modeling honeybee diseases using Python. Now, I knew a lot about honeybee diseases, but as a hobby I kept bees, and I'd already written quite a successful book on honeybees and regularly appeared on radio and television talking about honeybees. So the veterinary science department in Melbourne University were quite happy to take me on board to do a computer modeling project looking at the spread of diseases through honeybee populations. So the department I'm in is epidemiology. So epidemiology is the analysis of the distribution and spread of diseases in humans and animals, okay? To manage infectious disease outbreaks, governments need to understand how the disease will spread. Something like influenza, which is quite well understood by doctors around the world, we understand how that's going to spread if it appeared in Bangkok tomorrow. But a lot of diseases, say like Ebola in Africa, that are quite new, we've no idea how they're going to spread. So we need computer modeling to try and predict what's going to happen in the future. The problem with the spread of diseases and animal processes in general is there's so many random factors involved in it. The programs get quite complicated just because there's so many random events happening. How the disease spreads, how people catch it, how animals catch it, how infectious it is, what the weather's like influences it. So it's a lot of random processes which make the programming complex. So there are many thousands of ways the disease can spread. So how can we prepare for this? Experience, as I said, can only tell us so much. If a completely new disease like Ebola appeared, we have no idea how that's going to spread. And although Ebola had appeared before in Africa, when Ebola appeared in the United States, it threw everybody. No one knew how Ebola was going to spread through Texas, although they knew how it was going to spread through tropical Africa. So computer modeling helps us a lot. And computer modeling is used extensively these days to model biological and health processes, both in humans and in animals. So the model I use, because it's a stochastic process, I've run it 400,000 times now to understand what's happening. The models must be realistic to get reliable results, so it's a team effort. You need people who understand the animals in particular, in my case honeybees, but you also need experienced programmers to design the programs, because the programs can be quite complicated. Something like HIV/AIDS, which is quite widespread now, the programs which predict that are enormous. They're really, really complicated to predict how AIDS is spreading through the human population in the next 10 years. And getting it wrong can be very expensive. There's an animal disease of cows called foot and mouth disease, which appeared in the United Kingdom in the year 2000. They modeled the spread of that. The veterinarians who advised on the model got it wrong. They thought it would be much more serious than it was, and they ended up killing like half the cows in the United Kingdom, thinking it was going to be a serious problem, and it wasn't. So getting the model wrong can be a really serious problem. For honeybees now, I need to explain some background of honeybees. So the honeybees we're looking at, the so-called Western honeybee, they can either be kept in beehives, the so-called managed beehives, or they can live in the wild in trees, the so-called feral bees. They're the same bees exactly, but one is kept in a beehive by beekeepers, one just lives wild in the tree. And there are differences between them, okay? But remember the term managed and feral. And a colony of bees, and a colony of bees is about 40,000 bees in each of those. There's 40,000 individual bees in a managed beehive, and about 40,000 bees in a feral colony in a tree. Then we've got the terms colonies and population. There's an individual colony of bees on the left, either in a beehive or in a hole in a tree. And as I said, there's about 40,000 bees in that. And on the right is the population of bees. All the bees in Thailand would be the population of Thailand bees. All the bees in Australia, where I come from, would be the population of Australian bees. And they comprise both feral bees and managed bees. So colony and population are different terms. Bees have become critical to us recently in that in the past, say, 20 years, a large amount of them have been dying for different reasons. Diseases, as I'll explain shortly, is a big cause of death in honeybees, and why this is important, I'll explain. Last year in the United States, 28% of all the colonies died, and that has a big impact on agriculture. Some years in the United States, 44% of all the bee colonies died, and that actually is a staggering number, considering the background rate should be about 5%. That's in managed beehives. In feral beehives, feral colonies, shortly after this disease arrives in a country, 95% of all feral colonies are dead. Okay, so it's a really big impact on honeybees. Why are we concerned about this? 70% of all the foods we eat are pollinated by honeybees. If there's no honeybees around, we won't get any of these foods and a lot of other foods. So honeybees are actually really critical to our survival. So the fact that there's so many of them are dying is something we've really got to get a handle on. Why are bees dying? There's various reasons, but one of the main reasons is a small mite called Varroa. Varroa jumped species from other animals onto the honeybee in the 1950s. It happened in Korea. It started in Korea and spread around the world. So these mites are sucking blood of honeybees. Now, in itself, Varroa isn't a problem. It's like the mosquito. The fact that it bites you is a nuisance, but it's not really a problem. The problem with the mosquito is when it sucks blood out of you, it injects virus or diseases like malaria and yellow fever, and that's the problem. It's the same with Varroa. Sucking blood isn't a problem. It injects into the bees a virus one of them called deformed wing virus, which kills bees. And it's this transmission of virus by the small mite that's around the world. This is everywhere in the world now except Australia. Australia is fortunate, it's been free of it, but it'll arrive maybe this year, next year. It's been caught on ships, escaping quarantine, but they've killed it. But one day it'll arrive. And within 2 years of arriving, 95% of the feral bees will be dead, and that'll have a big impact on agriculture. I'll go on to now what we need to model in the Python programming. The way honeybees reproduce is called swarming. On the left, there's a gentleman there with one colony of bees. In that colony, there's one beehive. There's one queen who lays all the eggs and about 40,000 bees. And the colony expands in numbers. Every day, there's more bees in that colony. And there comes a time, maybe once a year, when there's so many bees in that one colony that half the bees decide to take off and find a new colony. And you can see them flying off in the middle picture. So in a healthy colony, but once a year, half the bees take off to find a new colony, form a new nest. They take the old queen with her, leaving behind half of the bees without the queen. So swarming is the way honeybees reproduce. Half the bees take off with the old queen, half the bees stay behind and form a new queen. And we need to model this. So half the bees, as I said, take off to form a new colony. Half the bees remain behind, but there's no queen there yet to lay eggs to keep the colony going. So the bees rear a new queen. And because about 2 or 3 days after she's hatched out, we call her a virgin queen, because at this stage she hasn't mated yet. Don't know what happens here. Okay, she hasn't mated yet. So 2 to 3 days after she hatches, she goes on a mating flight. And when a queen leaves the colony to go on a mating flight, she mates with 16 to 20 male bees in midair, about 20 to 40 meters up in midair. She returns to the colony having mated with 16 to 20 male bees and never mates again, never leaves the colony again to mate with other bees. And all this must be modeled stochastically. So the queen stores inside her stomach or inside her abdomen the sperm from 16 to 20 male bees, and there's 2 to 4 million sperm in here. If she's laying 1,500 eggs a day, over 2 to 4 years, she's going to use up that 2 to 4 million sperm. So all this needs to be modeled stochastically. Now, some bees are resistant to disease, and some bees aren't resistant to disease. Most of them aren't to start with. So the male bees, the queen flies off to mate with in midair. Assume in this example that 90% of the males are not resistant, okay? And 10% of the males she mates with, males there, are resistant. So she flies up, she randomly mates with 16 to 20 bees, male bees. She'll get some of the sperm will be resistant, and some of the sperm she'll get will not be resistant, okay? So, and the different virgin queens that fly up there mate with a different random selection of bees, so they're more or less resistant sperm. So when she goes back to the colony and starts laying eggs, the next 1 or 2 days later, assume that 90% of the sperm she's got is not resistant and 10% is resistant. Of the baby bees she lays, 90% will not be resistant and 10% resistant. So if 10% are resistant, how resistant is the colony as a whole to disease? What's the probability of death of that colony in 1 year? Okay, we don't know. If the figures are reversed, so only 10% are not resistant and 90% are resistant to the disease, again, what's the probability of death of that colony in a year? We don't know. Okay, there's a lot of factors we don't know in trying to model this disease. If it's 50% resistant, non-resistant, what's the probability of death in a year? All these factors are completely unknown to us. It's critical we get a handle on this. We can't measure it in the field. We've got to try and model it to see what's happening. So I'll go into the model now. I mentioned there's the managed population and the feral population in Thailand. And both of those produce male bees that fly into the sky, and all they do is hang around waiting for virgin queens to mate with. Okay, that's their sole function in life. Unfortunately, as soon as they mate, they die. They fall off dead. So I look at the managed population first because it's much easier. Managed populations aren't affected by diseases so much because beekeepers can add antibiotics if they see it being sick, or they can add insecticides to kill the Varroa. That causes problems, but not as much as Varroa itself. So when the going off all the time. So when a colony swarms, okay, first of all, a virgin queen leaves the managed population, mates with the bees in the sky, and goes back to the managed colony and takes over that colony, laying eggs and looking after the colony. The same with the feral population. When that swarms, the colony form a virgin queen. She flies out, mates randomly with male bees in the sky, and the males come from both managed and feral population, returns to the colony, starts laying eggs within a day or two. I mentioned that when colonies swarm, half the bees take off. So in the managed population, when half the bees in that hive take off, they've got nowhere else to go, so they join the feral population and fly off looking for a hole in a tree to form a nest in. So the feral population is continuously expanding. When the feral population swarms, the feral swarm goes off and comes back and joins the feral population looking for a hole in the tree. So the feral population is continuously expanding, whereas the managed population stays the same number roughly every year. And all this must be modeled. We look at the model for managed colonies first, because this is quite simple. In the managed colony, there's about maybe 10% of the bees are resistant to disease, because we can breed resistance in diseases. The next thing we have to model is the males produce the queen produces male bees that fly into the sky to mate. Okay? The male bees, some have resistance and some don't. It's all to do with the DNA, the genetics, the chromosomes of the queen. I'm not going to really touch on that in here, but modeling the spread of DNA in populations is quite complicated. Okay? So you have to look at the spread of chromosomes. So the colony produces male bees. They fly out into the sky. Some are resistant, some are not, depending on the resistance of the queen. Then some of the colonies swarm. The numbers build up inside the colony, and in the managed population, in a typical year, between 10 and 30% of the colonies swarm every year. Okay, it varies depending on the weather, the food supply, and then the cycle repeats itself. So managed populations are quite straightforward. The feral population now is more complicated. There's very little resistance in the start of year 0, the start of the program in the feral population. Maybe 1 or 2% of the bees are resistant. So the feral colonies produce male bees, the same as the managed. The colonies swarm. Okay, there's an increase in population, and then the next thing that happens is, over winter, because Varroa kills bees over winter, so many of the feral colonies are killed by Varroa. How many of the colonies are killed by the Varroa, or which colonies are killed, depends on the resistance. Depends on how many males the queen is mated with that has resistance to Varroa, will determine which colonies get killed. And there's a probability of death in that. Maybe 80% will die if there's very little resistance. So it's all stochastic processes. If there's a high resistance to death, high resistance to Varroa, maybe 10% of the colonies will die. There's a 10% probability of death in the colony, in that population. Now, the colonies increase in numbers every year. The population increases. But there's only so many colonies that the environment will support. There's only so much food out there. There's only so many nests feral colonies can take up. So once Varroas kill bees, if there's too many bees for the environment to support, say food, the environment's going to start killing bees by starving to death, or they may not find a nest to live in, and the next rainstorm comes along, drowns them all. So there's a lot of stochastic processes. And then at the end of the year, the whole process is just repeated. Now, the average resistance of the colony. If you look at the population as a whole, I split the population into 3 groups. If they got between 0 and 33% resistance, so if up to 33% of the bees in the colony have resistance, there's a high probability of death. It's up to 100%, but we don't know what that number is. If between a third and 2/3 of the bees in the colony have resistance, I had a medium probability of death. And if between 2/3 and all the bees in the colony had resistance, there's a very low probability of death. But we don't know what these numbers are. We know some factors. This isn't very good, is it? Okay, so we've got to stochastically model this. So the random processes are the percentage of resistant males the queen mates with. She goes up and mates with queens. That's a random process for every virgin queen that goes up there. The probability of death by Varroa, low, medium, or high. I randomly put figures in for that. That's randomly chosen. And I've run this program now like 400,000 times to get in all the combinations. Within the different resistance bands, which colony dies? If 10% of the colonies die, which 10% die? And I've randomly selected those out because that has an effect. Which colonies swarm? In managed population, between 10 and 30% of colonies swarm every year. So they are randomly selected out. In the feral population, if in a really healthy feral population, 80% of the colonies will swarm every year. But if the colony is diseased, maybe only 10% will swarm. We don't know these figures, so I have to allocate randomly numbers to see if we can replicate what's happening. Okay? Now, the DNA part is quite complicated. I don't want to go into this here, but when the queen goes off to mate, the male bees have resistance in their DNA and their chromosomes, and that's taken back to the queen and stored in her body. There's 2 to 4 million sperm that's stored in her body, which she uses up 1,500 times a day. The queen also has 2 chromosomes like humans, and when she lays an egg, one of those chromosomes goes into the egg, roughly. Okay? So then the female worker has 1 chromosome from the queen and 1 chromosome from the male bee. But it's more complicated than that. So that again is a stochastic process. It's a random process we have to model. Now, how did I do this? For the mating up in the sky, if 10% of the bees are resistant and 90% of the bees are not resistant, then I had a list 100 elements long. I had 10 ones in there and 90 zeros. 1 represented a male bee with resistance, and 0 represented a male bee without resistance. And when a virgin queen mated, I randomly selected 18, between 16 and 20 elements out of the list to represent the number of resistant bees she mated with. And this varied for each virgin queen that went up there. So each colony has a different resistance. Even they may have sent a virgin queen up to mate on one day, it varies from it varied from colony to colony. Okay? Now, deaths due to Varroa. So in Eastern Australia, there's about 500,000 feral colonies, we believe. We're not certain. On the day Varroa arrives and spreads, within 2 or 3 years, 95% of the feral colonies are going to have died. Okay? And as all the weaker colonies will die, and as resistance builds up in the population, the number of beehives, number of feral colonies increases slowly. So it's back to its normal population within 10 to 12 years. Okay? But after 10 to 12 years, the number of colonies, if I left the model run as it is, the number of colonies would increase beyond what the environment can support, beyond how much food supply is there. So I've got what's called the carrying capacity of the environment. And once the number of feral colonies out there reaches the carrying capacity of the environment, there needs to be a new killing process in there, which brings the number down to whatever the environment can support. And factors involved in this are quite random, like what's the weather. Poor weather could well increase the death rate. Good weather may not, depends on the time of year. If there's good weather towards winter and the bees remain strong towards winter, that means Varroa is going to build up very quickly over winter. So good weather towards winter means a higher death rate over winter because Varroa will be stronger over winter. So weather is impacting in this model quite a lot, and that's very variable. Okay, so these are some of the results. Look at the bottom chart first. The bottom chart. The two things we know are, within 2 or 3 years of the disease entering a country, the feral colony, the feral population completely collapsed, and it's rebuilt in about 10 or 12 years with resistant bees. If I randomly model in the bottom chart the probability of death for low, medium, and high resistant bees, if the probability of death is low, a low probability of death for all the resistances, the middle chart there shows a slight dip in the population of honeybees, but it's not a big dip. Okay, but that's not what we're seeing. So the probability of death from chromosomes isn't low if there's an infection. It's high. And the top graph represents the spread of resistant DNA, resistant chromosomes, as a percentage in the population. You can see the blue on top is the non-resistant colonies slowly decreasing in numbers over 15 years. The middle yellow band is the medium resistant colonies increasing to start with, but then decreasing. And the green on the far right is the high resistant bees increasing in numbers, but there's no big dip in the population. So this isn't correct. Stream, no matter what the resistance is in the population, there's a high probability of death. And you can see the number of bees in the middle chart catastrophically declines in the population, but never recovers. So the probability of death over low and medium resistance is certainly not the situation we are seeing in nature. But if you look at this example now, if there's a low probability of death, if there's a low probability of death if there's a high probability of death for low resistant colonies, and then quite a small probability of death for medium resistant and high resistant colonies, the population declines rapidly in 3 and 4 years, and then increases by year 11, 12. So this is what we're seeing in nature. Field studies in France and Sweden and the United States have replicated this exactly. So we now understand better by mathematical modeling with computer exactly what the parameters are that cause the replication in nature. And by year 12, the population size has increased above what the environment can support. And you can see the blue bars on top are the colonies being killed by lack of food. So we understand now the relationship between resistance in the colony and the probability of death. We're much closer to doing that. Now, for the data structure I used, for every single colony out there, I had a 10 list data structure which represented the key parameters of that colony. If I had 450,000 managed colonies and 450,000 feral colonies, at the start of the program I had 900,000 of these data structures. As the colonies swarmed and increased in numbers, by the end of 15 years I had several million of these to sort through. Okay? That's for each colony. For the population as a whole, I had another 10 element structure, 10 element list, listing at the start of year 0 and at the end of every year to year 15. So I had 16 of these lists specifying the key characteristics that I was interested in at the end of each year. And at the end of the 15-year simulation, I had a 25 element data structure as well. So there's quite a bit of data in this. On my laptop, which is quite old, about 3 or 4 years old, to run this 10,000 times took about 4 hours. I could run it on a supercomputer in the university, but it's easier just to run it on my laptop and leave it go. I'll go out for a coffee. Okay, now, so this is typically what's happening. By randomly varying everything and running this population, we've managed to replicate and understand much more clearly some of the key parameters involved in the death of honeybee colonies. Okay, so we now know that what we were hoping to see was that if we put resistant bees into the managed population, and we can breed resistant bees quite easily, there's people who do that, that they'll swarm into the feral population and increase resistance fast in the feral population. But we're not finding that. What we're finding with the model is, no matter what we do in the managed population kept by beekeepers, it's not having an effect at all on the feral population. We've just got to let the disease run its course in the feral population. It'll affect agriculture. If we didn't do anything about it, there wouldn't be enough feral bees around for us to get apples and pears and blueberries and mangoes or whatever. So we've got to significantly increase the number of managed bees, like increase it by 50%. And so we understand now much better what we need to do for managed bees to support the feral population, to support agriculture. Okay, so in conclusion, modeling can be used to simulate different aspects of a disease. It's used extensively in medicine, in veterinary science. It's used in knowledge of genetics and chromosomes. So you can understand the decline and recovery of feral bee population, but any disease like flu, AIDS, SARS, it's used extensively in universities. There's a lot of programmers out there working for universities and large hospitals. It prepares us for a collapse of the honeybee population and can be used to simulate any disease, and it's used widely and it's a very useful tool. There's a lot of Python and C++ programmers out there working in this area. Okay, so humans are reliant on bees for food. If the bee population collapsed, it'd be pretty bad news for us. So bees are reliant on humans to keep them alive, and Python programming and epidemiology and modeling what's going to happen in the future with the diseases is critical to our survival. Okay, thank you. Any questions?
The spread of SARS was largely affected by the level of travel people doing actually on planes and things. Is there is that sort of geographical factor?
In this model here, there's no geographical spread. But in the model I'm working on now, which is in R, not in Python, in R, it is modeled geographically. So you get a map of Australia, and you can see the disease spread. So we are working on an R model to look at the geographic spread and incorporate this. Yeah. Okay. Yep.
Specific Python packages, or is everything custom code written from scratch?
It's all custom coded. Now, that's because I don't know Python. I think if you guys program this, you do a lot better job than me on it. Okay. You know, so I taught myself Python recently, and so a lot of the advanced techniques I wouldn't know about. So it's just easier just to do it. Yep. Well, no, I do use packages. Yeah, it's not. Yeah, in a sense, I pull things out. Yeah. Okay.
I continue the question with Alex. How do you compare with another programming language? I mean, have you tried like using C++, and why you choose Python?
I use C++ because it's the quickest. I programmed in C++ before, but it was a long time ago, and but so I just chose Python because it's quick to pick up and easy to learn. In retrospect, I probably shouldn't have. Not anything wrong with Python. I should have used Java because all the academics in university know Java, not Python. So when I talk to my supervisor and other researchers, they only know Java, not Python. So.
Do you still use Java for the computational performance?
Do I use Java? I don't use Java at all, no. I only use Python. But the university as a whole use Python. So it was probably a bit of a mistake me using Python. But it's a good program, though. A lot of people use Python, so. Okay?
What type of model framework do you use? Is it individual-based, compartmental? What sort of structure?
Well, I just got individual beehives and model individual beehives. Each beehive, I assume, with the same number of bees in it, but it has different characteristics depending on the genetics of the queen. The queen is individually modeled in each beehive, and the 16 to 20 drones, they're quite easy to model because they just have. But the queen is more complex to model the genetics in her. Yeah. Okay. Okay. Thank you very much. Oh, question here. Sorry.
What kind of simulation techniques do you use for this one? What kind of simulation techniques do you use for this one? Do you take into account all the factors you mentioned?
Sorry, I'm a bit deaf and I'm missing that. Say it again.
What type of simulation techniques do you use for this one?
Oh, I just put a random processes. I just got a random number generator in there. I specify an upper and lower bound in it, a standard Python thing. And when I run the program from through a year, say swarming now. Swarming for managed colonies, between 10 and 30% of colonies swarm every year. So at the start of every year, I just use a Python program that pulls out a random number between 10 and 30, and then I randomly chose, say it's 20% swarm, I randomly chose 20 colonies out of the managed population. I just pull random numbers out. It's not rocket science what I do in it. But the hard bit of this isn't so much the programming, it's modeling the biological model. You know, in this, weather isn't taken into account, but in the R model, where I'm looking at a spread geographically, weather is taken into account. I'm randomly applying thunderstorms or the frequency that apply in Eastern Australia generally. You know, the random processes aren't hard to model. You know, it's getting the biology correct. Okay. Okay. Okay, thank you.